Hi, folks. Thanks for coming. Most of you know me. My name's Adam. I lead the data science and sharing team. I'm talking to you today about data sharing and open science. Just real briefly, just give you, there we go, a roadmap of the talk, which is basically a bunch of questions. Question number one is, why doesn't my mouse work? There we go. Question number one, who am I? What does the DSST actually do? Most of you guys know me. There's some folks on the Zoom that don't know me yet, or at least I don't know them. Why do we need a DSST and what problems are we trying to solve? And then assuming that I answered those and convince you, the third section is that you are convinced and what you can do to help. So starting off with who I am and what does DSST actually do? This is the DSST. We are a team of 6 people. Dustin Mortescu is in the audience. Eric and Mia, and I think Arsh, maybe Arsh, yeah, are all on Zoom. And we're here to help you with your data science and sharing needs. Briefly, this is our mission statement, which you can find from our annual report, which is linked from our website. Our goal is to support and advance the creation, distribution, and use of open data sets to accelerate discovery within the NIMH IRP, as well as the NINDS IRP. We have some NINDS representation here. We provide tools and training to help scientists embrace open and reproducible science practices, which include open distribution of code, data, methods, standardized formats and data repositories, as well as a culture of transparency that is focused on identifying and correcting errors quickly. We'll get into that a little bit more. But if you want to know what we do in more detail, we had our Board of Scientific Counselors review for the newer trainees. All NIH intramural programs go through a quadrennial review with the Board of Scientific Counselors every 4 years. And our entire review is there. It's just 10 pages. It's available, that link at the top, and you can read in lots of detail and about 110 different references about the kinds of stuff we've been up to for at least the past 4 years. I'm happy to report that our BSc went well. We got a very good score, which helped us justify the addition of one more slot. We will be welcoming Josh Lorimore tomorrow, who is coming to us from the world of microscopy and pathology and a bit of sequencing as well. So we are expanding our mandate but beyond sort of traditional neuroimaging with Josh. Another bit of news that I'm going to debut here is that Jessica DeFlan, who has been on our team for a while, working closely with the machine learning team, has accepted a position with Valence Labs in London last month, where she's going to be doing some really interesting machine learning stuff on drug generation or drug discovery, I should say, which leaves us with an open slot. So we are recruiting. This is probably going to hit Twitter a little bit later this week with the advertisements. But if you or anyone you know thinks you might have a place on the data science and sharing team, please let us know. The ad is right there on GitHub at that link. And these slides will be made available through the neuroimaging website. All right, coming back to the roadmap. Why do we need a DSST? What problem are we trying to solve? And there we get our first poll. What percentage of peer-reviewed papers in the biomedical literature present results that can be replicated by another scientist in the same field? So folks in the audience, you can just raise 1, 2, 3. Folks on Zoom, if you would please respond with the emoticon about your guess. I got 3. I got some abstentions. No? Oh, 3. 1, 2.
Or 3, but definitely not 3.
All right. We got 3s or 2s, and the correct answer is less than 50% to whatever degree that we can estimate it. Some of you may be familiar with the paper that kind of started all this, the replication crisis from Ioannidis, which included some estimations in there. There have been additional estimations that have looked at actual chunks of the literature from psychology, from cancer, and tried to reproduce them. And all of those numbers, this one comes up from Cancer. 47 of 53 studies could not be replicated, which puts us much closer to sort of the 10% range. And there are a number of papers out there that have tried to answer this question, and all of them are solidly below 50%. This is another example from psychology where they conducted replications of 100 experiments, and again, the replication was was very low, and even the ones that did replicate them, the effect sizes decreased in magnitude substantially. Okay, I got another poll question for you. Papers published in prestigious journals are more likely to report reproducible findings. We got a lot of twos in the audience from the group. I see, yeah, falses. Okay. Oh, wait, no, we got one person who thinks that the prestigious journals are more reproducible. So the answer is indeed false. When they go and they look at these individual studies about the ones that are reproducible, if anything is anti-correlated, the highest impact factor or whatever measure of prestige tend to be less reproducible. Yeah, this is again coming back from that cancer study from 2011, is that they do not correlate with journal impact factor at all. Poll question number 3. Since Andy's explosive 2005 paper, the field has become more vigilant, and the number of unreproducible findings published in the literature has decreased. True or false? False. 2 falses from the audience. From the Zoom. Oh, you're doing the icon. That's great. Because you're right. I didn't give any numbers here. That's right.
You're doing the number there.
Okay. So maybe it's getting better, but if it's getting better, it's getting better slowly. We are seeing sample size evolution in neuroimaging research. So if you evaluate the highly cited studies and the latest practices, we are starting to see larger samples, and as we'll discuss in a minute, larger samples correlate with more reproducible studies. Another thing that correlates with reproducibility is transparency. If you focus on these lines at the bottom, they show data coding and data sharing and code sharing, and they are creeping upward. But as of 2020, we're still around 15% for code sharing. But it is still happening a lot. So you'll still see studies like this one. This is a popular press coverage from Ed Yong that was published in the Atlantic a few years ago that talks about this particular SNP that was correlated with depression, that became the focus of a whole lot of depression research and a great deal of research money. And it turned out that when we actually got a large enough sample size and looked at it, it was simply something that was in the noise. But it was identified early and everybody focused on it for a very long time. So people think of the reproducibility crisis as something that is over 20 years old. That's true, but we have not yet solved the problem, which is why this is something that I continue to harp on and why groups like ours exist.
It's not just sample size, right? If you don't mind.
Yes, sorry, absolutely. Sample size is not the only thing.
Larger sample size, you also can be trying to detect smaller effects. So
yes,
sample size itself is not really the issue.
We'll get into that a little bit in the next section about sort of like what are the contributions to the reproducibility crisis. But absolutely, sample size is not the only thing. Sorry, this breaks down what I just said out loud. The 1996 study of 1,000 subjects claimed that a candidate gene was found to influence a person's risk of clinical depression. And 17 other candidate genes were subsequently linked. 1,000 papers were published on these genes, but a 2019 study showed that none of the 18 genes were related to depression risk any one chance. And the lead author said, how on earth could we have spent 20 years, hundreds of millions of dollars studying pure noise? And this all comes down to the things that lead us to the reproducibility crisis. Poll question number 4. Scientists are pretty good at predicting, aka guessing, which findings would be reproducible in published papers. So again, 1s and 2s from the audience, or yeah, you can do thumbs if you want. We got false, false, false. On the Zoom, everybody's saying false. I got you on this one. It is actually true. Well, to the degree that we've studied. Again, we have these studies where people have gone and taken 100 papers and they've tried to see if they are reproducible. But once you have that, once you have ground truth, then you can go back and you can poll scientists, like, which of these do you think are reproducible, just by looking at them. And they can, so this is one thing where they've looked at what correlates with various results are predictable. They do pretty well. Scientists are able to look at things and we can actually ask them the kinds of things that they look at for those predictions. So let's just to polling question number 5. What features of publications correlate with findings that can be reproduced? So we're going to pick just one of your favorites right now. So we got 1s and 5s for the audience. And we got an array of 5 different emoticons for the folks on Zoom. Number 1 is large effect size. Number 2, published in prestigious journals. I guess we've already kind of given that one away. Number 3, large sample size. Number 4, well-respected lab or institution. Or 5, transparency, availability of data, code, and methods. We have a 5 and 1. The folks on Zoom seem to be going for the transparency. I mean, that is the theme of the talk, so I think that's a pretty good guess. Okay, and again, so this is something that we can now sort of do science on it. This is something that came out in PLOS One a few years ago, predicting the replicability of social science labs that can parameterize it. Again, we're in social sciences here, so this is a subset of what the reproducibility problem affects. But as Peter correctly predicted, sample size is a big one. Large effect sizes as well as sample sizes are things that are going to predict reproducibility. That's one of the things that scientists pick up on. As we've already discussed, published in a prestigious journal is not something that correlates in any way. For the other ones, well-respected lab, institution, or transparency of data, code, or methods, the transparency is a big factor. It's being recommended in papers like this one about such a manifesto of reproducible science. Also, the well-respected lab institution is a little more complicated. So isolated and siloed researchers are more likely to produce irreproducible results because they lack the many eyeballs that we get from transparency. However, high-ranking labs may be blindly trusted or subject to less scrutiny. So you can actually get the opposite effect. If there's a whole lot of respect, perhaps no one has actually looked at it before. And I could tell a lot of anecdotal stories of that happening. Also, groupthink is surprisingly common in science. Creative out-of-the-box ideas can come from labs outside of the establishment. All right, so summary of this part, things I've told you so far. The number of unreproducible findings in the biomedical literature remains alarmingly high. Some irreproducibility, it's a hard word to say, is just part of science, but the majority of it is preventable. There has been some progress in changing the norms and behaviors, but it's been moving very, very slowly. Any questions in this space before we sort of go on to what we can do about it? Both from the room and from the Zoom. I guess put it on chat if you have it on Zoom, or if you want, you can just unmute and shout out.
So an ideal case, if everybody plays by the rules and does the right thing, what percentage of work would you expect to be still irreproducible?
Let me just repeat the question. If everybody was doing all the right things, what percentage of the work would be not reproducible just by the matters of science? I don't immediately know the answer to that question, but there is a body of literature that addresses that.
If you just set a certain p-value and you say, well, you report an effect that happens to be significant, you still have a chance.
Right.
It turns out to be wrong. If you repeat the study with 100 times more people, and you have a more sensitivity, and you find out it was noise after all. So
right.
There is a certain
right. And so what that number is going to be somewhat dictated by the p-values that you're using and whatnot. So it should be somewhat predictable. But I think we would also expect that there are going to be factors that people aren't reporting. So Stuart Buck, who runs a really interesting Substack and a project called the Good Science Project, he tells a story about particularly high impact finding that they were trying to reproduce and failing. The labs were collaborating. And they spent years and lots of time trying to do the exact same thing in 2 different labs until they finally just had to put the people side by side and analyze it step by step. And it turned out to be that the difference was the rate at which the solution was being stirred. One side was stirring it very quickly. The other one was doing it slowly, or actually it might have been an automated device, but like surprisingly subtle things can lead to a lack of reproducibility. And you can't. It will be difficult to get to the point where we report all of those things. So a certain percentage is expected, but certainly I would hope that it's less than 50. Anyone from the Zoom? Oh, there's a chat. Oh, that's just messed up. Okay, I will jump into the next part. All right, so let's say I convinced you, and what can you do to help? So again, this is largely targeted at the trainees out there. I kind of see this course as focused on the postdocs, postbacks that are in the audience. But this applies for staff scientists as well. And the subtext question is, if all biomedical science has been mired in a reproducibility crisis for nearly 20 years, is there anything a data science-minded NIH trainee can do about it? And I guess I would say emphatically yes. So our team, if anything, we try to target the trainees because we believe that is where the change is going to come from. The scientists who have been doing this for 20, 30, 50 years are largely happy with the way they're doing it, and they're far less amenable to change, with notable exceptions. But in the trainees, we find that trainees is where the change comes from. In fact, the trainees are the key to fixing the mess. So how can you improve your lab's reproducibility? So here's a list of a couple things that I want to talk about: pre-registration, planning for data sharing at the beginning of an experiment, version control and code sharing, and that basically comes down to Git and GitHub, dealing with errors in your code, which includes unit tests and continuous integration, which we'll talk about a little bit. Making your code reusable. I took out the bit on containers to save some time, and reporting and dissemination, which is preprints and checklist. So first off, pre-registration. I feel like this maybe is possibly the most controversial, and maybe isn't going to make sense in every case, but our lab has had some great experience with it. It is the simple process of making a record of your experimental predictions. So some journals now have a process of sending it in and having that pre-registration reviewed. But you can do something far simpler with websites like OSF. It doesn't prohibit or prevent you from doing exploratory research. You just have to clearly define what is exploratory and what is hypothesis-based. It does help the investigator and the reader of the journal, of the output, distinguish between predictions and postdictions, or HARKing. HARKing is an acronym for hypothesizing after the results are known, which happens explicitly and subtly in science, I think a lot more than most people realize. If no one ever wrote down what the hypothesis was, it's very easy for it to slowly morph as each bit of data comes in. And pre-registrations also combat publication bias. Publication bias being that we only publish the positive results, right? We don't say when things didn't work out, which leads to a lot of researchers doing the same field experiment over and over again. If it's pre-registered, then you report the results as to what didn't work. Planning ahead for data sharing for human subjects, you need to ensure that your protocol has acceptable data sharing language. There is lots of that available online right now. Open brain consent, readthedocs.io. You can find examples of language that people have put into their various consent forms from all over the world. Varies a bit from country to country, but if you have a bit of text that has gotten past one IRB, then it's more likely to get past the next IRB. And when designing and collecting and analyzing, you can consult with standard documentation. There's a whole lot of, depending on what kind of data collection you're doing, there's a whole lot of checklists and various other best practices documents out there that can give you guidance about what you need to think about. One is Equator, which stands for Enhancing Quality and Transparency in Health Research. That's quite broad. There's best practices in data analysis and sharing in neuroimaging using MRI, which works out to Covitus somehow. That's mostly about reporting, but I think as you read through Covitus at the beginning of your study, you think about the things that you need to choose explicitly and report in the paper. And also at the beginning, you can think about which repositories might be best for your sharing of your data, what structures and formats it will require. If you're thinking about that at the beginning of the study, it's far easier than doing it at the end. Sorry about the font color change. So version control is another thing that our lab spends a lot of time teaching, training, and talking about. That basically amounts to GitHub, or alternatively, there are other platforms like GitLab or Bitbucket. Version control systems allow you to store all of your analysis in a central repository and keep a history of snapshots of your evolving analysis, which again, it sort of gives you a map of how things diverge and progress. You can also quickly switch between different versions of your analyses. You can adopt and modify code from other scientists, and you can collaborate. Again, this just sort of brings multiple eyeballs into the problem. It creates a system by which you can have other people look. And if you want to take it to another level, you can also do code reviews. Modify your code. You can create what's called a pull request. You can invite people in your lab or people outside your lab saying, hey, will you review this pull request before I incorporate it into my main analysis code? This kind of thing is routine in the world of engineering and especially software engineering, but it is still relatively rare in the sciences. That's why the covers are off. And we have another poll question for you. I forgot to put the icons in here. Oh, wow. All right, we'll just go across the ones in order. Studies have shown the computer code written by scientists has an average of less than 1 defect per line of code. Number 2, 1 to 10 defects per 1,000 lines of code, 10 to 20 per 1,000 lines, or greater than 20 defects per lines of code. I got 4s in the audience. You guys can just, the folks on chat, you can just type in numbers into the thing. I see some, these are the recent ones. I see some 3s and 4s. Oh, Eric is optimistic with 2. Yeah, you're right. The answer is number 4. So they have looked at this. One 2003 study looked at coding errors from professional software developers, and they make an average of 1 to 7.5 defects per line of code. And these are people who are trained in software engineering who do this all day long. They are still making as many as 7 errors per. Oh, I missed 1,000. Sorry, per 1,000 lines of code. There's not an error. If there was yeah, you wouldn't get far if there was an error in every per 1,000 lines of code. That's other estimates with the number quite higher, at 10 to 50 defects per lines of code. So it's really not a question of whether your code has errors. It does. It's a question of when you find them and if they and how they impact your results. Russ Poldrack has some great blogs about this. You are much more incentivized to look at errors that invalidate your results than errors that make your results more valid. So you always have that bias. So odds are, if there are errors, they are errors that make your analysis not actually true. Sorry. So the typical approach to code testing in science, I think many of you will be familiar with. You write some code to do an analysis, you run the code on some of your data, and you debug the code until the output looks reasonable. Oh yeah, that makes sense to me. This is not the way software is developed in the rest of the world, ideally. And then obviously you can repeat this process as many times as you want. And this really calls me back to the Richard Feynman quote. The first principle is that you must not fool yourself. You are the easiest person to fool. As soon as you see something that is reasonable, which is going to be highly affected by what your hypothesis about the data is, then you are going to stop debugging, which means the bugs you're going to miss are going to be the ones that create false positives. There's another way to go about this, and it is common in the world of software engineering, and that is introducing tests into your code from the get-go. So as you write the code, you are also constantly thinking about what are the sanity checks you can do for any given function, for any given loop. And then you can write little statements using tools like PyTest to say, if everything's going well, then this should be greater than that, or there should be this many lines after here, or this should equate to that. And training yourself to include these is something that is not happening enough in science, but it's something that we are starting to push more and more with the stuff that we run, and it is really a pillar of the reproducibility community. For the folks that are just starting their coding journey as NIH trainees, I think this is something that I have to get in early on. Another example of testing. A basic PyTest tutorial is available through Russ Poldrack's website. You can also do something called continuous integration, where you ask GitHub to run the test every time you upload a new version of that code. And you get little flags that come up that passed the test or didn't. So that sort of takes some of the weight off you and also makes it easy for everyone else to see that your tests are passing. And you can also automatically assess how much of your code has been tested with a Python module called coverage. And this is not just Python. Unit tests are available in R or whatever language you're using. Testing is it's a universal thing that can be done. And then dealing with errors. So again, even in the best labs, sometimes errors will still slip through. There's an article here, it goes all back to 2006 about a software problem that led to 5 retractions. I'm just going to read a little bit from this. In 2007, Shang and co-authors retracted 5 previously published papers describing the structure of 3 multi-drug transporter proteins after another research group published a widely differing structure, which led to the discovery of a critical bug in the Chang's custom software tools. Again, I referenced this before. Russ Poldrack has a great blog article about how they also had an error when it was in a preprint that they had posted. And they described some issues that they had identified with the stop signal task in the ABCD study, and they published the code. And then the ABCD stop signal team performed a detailed review, and they notified us the error, and then they corrected it in the preprint before it actually went to publication. And then that eventually went on to become an ELITE paper. Oops. So the point there that I think was made well by Russ's post is that sharing code is critical for discovering errors. You do not have the eyes to discover it yourself. So you don't want to let embarrassment about ugly code stop you from sharing. And it's also good to own your mistakes, and do what's necessary to correct them. A good PI will applaud you for it. It's much better to find it in the preprint stage as opposed to after it's gone out to publication and you have to deal with retractions. The other thing that our team is working towards is trying to make errors and correcting them is part of science. It's not something we should be ashamed of. Again, it's happening to everybody. We just need to bring it out in the open. Normalizing errors is a critical step in addressing the reproducibility crisis. And we also encourage everyone to never mock or jeer another person's code, especially, and we all know that scientists produce some really ugly code that can be really hard to parse. That's okay. It's part of the process. So check yourself if you ever find yourself mocking someone's code as being sloppy. So coming back to preprint postings, so the Poldrack story, I think, is a really good illustration of this. If you put your results out on a preprint server before or while you submit it off to review, you immediately get a lot of extra eyeballs to look it over and find things. It is immediately open access, the opportunity to catch errors, and you've also put a timestamp in the public record. So if someone does choose to scoop you, you can always still cite the DOI associated with your preprint to demonstrate that you had the discovery earlier. Earlier precedence prevents scooping, and it also can speed and improve the final submission. I'm seeing a lot of people that now submit it to the journal and put it on a preprint server at exactly the same time, and it does seem to make things go a bit faster. I had mentioned the standards about Equator and Corbidus before, so that's a bit of redundancy. Okay, so take homes from that section. A large fraction of people listening to this talk will or have authored an unreproducible finding, which is bad. Some of reproducibility is just part of science, but a much larger fraction is preventable. You can influence yourself, your primary investigator, to do more reproducible science. You can make a plan for data sharing before you start. You can ask your PI to pre-register the study. You can make your code available, readable, and rerunnable. You can plan for coding errors and build in tests to catch them. You should not be embarrassed of your code, and don't mock others who have quote unquote sloppy code. You can report your work thoroughly using checklists and broadly using preprint servers. And also, we're here to help. The whole team is to help make these things happen. So if you find that you're taking on these things or finding them too difficult, reach out to us. That's our website. The email address goes to our entire team. We would love to help you, whether it's a neuroimaging problem or something broader. And that's the team. So that's about 30 minutes. So let's take some questions there. I have plenty of extra slides and other stuff I could talk to you about with the remaining time. But let's see if there's conversation on what we have so far. Anyone on the Zoom? Just chat. All right, the additional slides I included, so some of it is just kind of an update of stuff that we've been working on since our BSC. But I gathered that Jaco and Peter were here to talk a little bit more about the NIH data sharing policy, which I somehow managed to not mention. One of the reasons that you're hearing a lot more about data sharing these days, many of you on this on the call will know this, but the NIH issued a data sharing policy. Have a slide for that. They issued a data sharing policy that went into effect in January of 2023. So as of January 2023, everyone who gets NIH money is required to share their data, or at least to submit a data sharing plan. And that affects both intramural and extramural. Like all things with NIH, the implementation looks a little bit different for each of the different institutes, and sort of what makes sense and what is plausible. But there is a new press towards sharing data and being compliant with that policy. And we're helping a lot of investigators in drafting their plan and sharing their data, as well as to providing a level of evaluation. So we look at the papers to see whether there is evidence of data sharing, so we can provide reports to leadership. So it's something that we've worked on a lot. Do you guys I know that's more what you guys were talking about. Do you have any questions in that space?
Yeah, practical terms, better say just did a study, a bunch of subjects with marginal images, and basically now what? Right, there is no given data sharing plan to say, okay, we're going to share the data. Well, there is no decent repository where we can do this easily. So essentially, what do I do? How do we share a bunch of non-standard images that don't really fall into standard EPI, fMRI category or standard
Gotcha.
TT2 Siemens
Right.
XYZ scan, right? So,
Okay, let me repeat the question. So just finished acquiring a bunch of data, MRI data with a bunch of subjects, and that there is not a good repository choice. So what I know, which I think maybe people don't know, is that the physics group in NINDS does a lot of custom sequencing. There's very little that you guys do that is out of the box. So neuroimaging in general, I think, has more data standards than most other domains of science, with the possible exception of the genomics folks. So if you're doing, your bog standard Siemens, GE, Philips sequence, you can take your DICOM and run it through a DICOM to BIDS converter, and you can share your data. But if you're doing something significantly different, which you guys almost always are, that's not going to work. So there's a couple things that you can do. If you want to upload it to a BID-centric repository, so that could be OpenNeuro or something similar. There are ways to bend BIDs to your whims, right? You can always add a BIDs ignore file, and you can basically say, okay, well, this is not following the BIDs rule, but here are the files that you need to understand and possibly reproduce what we do. So you can, if it doesn't have certain aspects of things, you can always do that. And there are a couple of directories within BIDS, like the source directory, the code directory, and the derivatives directory, which are relatively unconstrained. So if you make a reasonable determination of what you think needs to be included, you can put it in there in what is increasingly becoming sort of a BIDS-inspired structure, right? Like, there's sort of there's a theme of basically doing subject session modality. Right. So a lot of people are just naming things like that, even though they haven't yet written a BIDS extension, they can do something that's very BIDS-like. So you could do that. And if that proves too much of a hassle or too tedious, like if your data is really so far away from BIDS that it seems farther away, you can always use what we're calling now a generalist repository. So the Office of Data Science just funded 5 of them, generalist repositories. So I'm not going to remember all 5, but Figshare is one of the bigger ones. Zenodo, Vivily, Harvard Dataverse, and there's one more, Dryad. And so they will take any data. right? So you could give them a tarball, you could give them data in any format. They will, most of them will run it through some level of curation, and you'll get feedback about metadata that is or is not there. But you're actually not required to listen to it, and they are not going to turn you away. So there's places you can put it and basically not follow any rules. Everybody in the data sharing world will say, I mean, there was a while where NIH wouldn't even report on these because they really wanted people to find a standard for their data, right? But shared data is better than not shared data. So if it really doesn't fit, then I think Bigshare or DryX is a great choice. We've definitely helped a lot of people with data that does not fit into a modality into some of these repositories, especially in places like microscopy world, electrophysiology. There's a lot of domains where the data standards aren't there yet. I got a question from Arshifa, who's on our team. Do you have any advice on at what stage of a research study folks can reach out to us being the DSST? And it's earlier the better, right? Like I think, and it doesn't have to be just one time, but this is something that Francisco and I lament about a lot. Francisco Pereira leads the machine learning team, who we share space with and work with pretty closely, is that we get the calls too late, right? For Francisco, it's about planning the analysis and planning, what kind of questions can be asked. For us, it's sort of dealing with the data collection, and then having to do those things post hoc. I think is far more difficult than doing it at the beginning. And then I think I've seen other things where they come to us at the very beginning, even maybe before the protocol's been written, and we can have a very high level conversation. But then as you're writing protocols and getting things together, things get busy. It's good to check in with support teams like ours and the machine learning team early and frequently. Like you can sort of like set up a periodic meeting like once every 2 or 3 months. Like as things evolve, you can sort of think about how is this going to affect the data sharing, how it's going to affect the plan analysis. But the earlier the better, and we're always happy to check. Other questions from the Zoom? Other topics you guys want to hit on as far as the data sharing space goes? Portachill. Do I want to launch into some more slides? Yeah, sure, why not? It's fun to talk about. So, where did I leave off? Sorry, I was looking at the other slide. I lost myself. So these are our take home extra slides. So most of what I talked to you about is up to our BSc and more recently. So in our, for those of you who came to our Board of Scientific Council's presentation, it included this slide. So we have been combing through the literature. So all labs report their publications in what's called the NIDB, the NIMH Intramural Database. So we scraped all those publications for full text to look for what data sharing actually exists. And you can see that in the blue and green dots. The red line is exactly the line from the paper that I showed earlier that shows when you look across all of PubMed Central, some 2.7 million papers, the data sharing rates are still close to 15%. But when we look across the intramural program, all of the intramural program, it's kind of on par. We only have 2 years where we're sort of comparing like to like. So the overall intramural program was just below it in 2019 and 2020. And NIMH, we were pleased to see was, NIMH was just below and all of intramural was high. But just in 2023, we've seen NIMH bump above the average for the intramural program, which we were very pleased to see. But we think that this is a way to sort of create incentives by actually looking at the literature and seeing where that reporting is. So I have spent the summer in Montreal. I'm actually just back on Saturday. I was what's called their Open Scientist in Residence. There is an organization there called the Tenenbaum Open Science Institute, which has put a lot of funding towards various open science initiatives. And I was collaborating with my colleague Jean-Baptiste Paulin. And the proposal that I sent to them before I went and did this little residency was to create something called the Open Science Metrics project. I worked on this with John Lee, who was one of the original employees of the data science and sharing team, who now runs his own company called Python AI Solutions, as well as another group of software developers called QuantSci. And what we proposed to the Tenenbaum Open Science Institute, or CHOICE, was for the Open Science Metrics project, was basically a set of 4 deliverables. One, that we would aggregate and harmonize the existing Open Science Metrics data sets into a single public database where people could access it and do metrics on it. We were going to create some simple tools to apply existing metrics to PDF publications and upload the results to the public database, as well as create a web accessible dashboard to allow visualization and to improve Open Science Metrics using LLMs. There's a bit of a piece that is missing from the intro. So this graph that I've been showing, this is produced by mostly by a freely available software program called OddPub, which goes through the full text of any given paper, and it does a relatively simple regular expression approach to look for URLs or particular repository names. It tries to figure out whether or not a given paper contains a data sharing statement or not. It's not perfect. In our hands, when we compare it to manual labeling, it's about 90% correct. If it's 10% error, kind of errors goes both false positives and false negatives. But it's a pretty good way to analyze 2.7 million articles. So this stuff is out there. This is one of the tools. There are other stuff out there, SciCrunch, SciTrans, and as well as this is embedded in something called RTransparent. But those tools are out there. So our proposal was to integrate them. I just gave this presentation to the folks who funded this a few days ago. And we largely put it together. We were basing it on another project that we've been involved in called MRIQC. So MRIQC is another freely available software that you can download on GitHub, and you can point it at, instead of at a paper, you can point it at a given MRI image, whether it's a structural image or a functional MRI image. It's basically in BIDS format, and it will go through and it will run a whole suite of various quality assessment metrics on it. And when it runs that, it will produce a nice report for you, like that web page, but it will also upload it to a database, the MRIQCDB, the web API, which we maintain here at NIMH. It's mriqc.nimh.gov. We've been maintaining it for, gosh, probably 6 to 8 years now. It's got a few million records in it. So if you want to go through and look at what quality metrics are like for the larger world, you can easily download this or query it and get whatever information you need. That's just a little bit more about the statistics on that. So we decided to use the same approach for the Open Science Metrics. So again, creating the database itself, this database now exists. It's in MongoDB, which we maintain. It has 2.7 million records. Again, this is all the stuff that was in PubMed Central as of 2020. You can now query that database through a REST API that is available at opensciencemetrics.org. And for the second deliverable, we also have a command line utility. So you can download this, you can do a pip install, and you can run it on any given PDF. Right now, you hand it a unique identifier like a DY or a PubMed ID, but in our next version, we'll be able to scrape that directly out of the publication. And then it will run the metric against that PDF and determine first it converts the PDF to an XML format, and then it searches the text for these indicators, and that goes up to the database. Here's an example that I just gave to the funders. This was a recent paper that had come out from the folks up in McGill. So I downloaded the PDF, gave it a reasonable name, ran this on the command line. It spins up a couple containers because some of these tools need to run in a containerized environment, and it finds a data sharing statement, and then it uploads it to the database. So this is the data availability statement, which you can also find in the PDF, unique identifiers and whatnot. So that is the first two. And then for the third, the dashboard, if you go to opensciencemetrics.org, you will find this dashboard. It is a little slow because it's dealing with 2.7 million metrics. And it's a little fragile, but it does exist. So the line that you're seeing right there is the exact same line that was in the previous figure that I've been showing, which just shows data sharing across the entirety of PubMed Central. But we have been focused on breaking it down by various kinds of classifications. So again, that's the same line side by side. So here we're breaking it down by country. Right? We can break it down by Canada, United States, France. There are dozens of other countries in the database that you could add to that. You can select them individually. But in our original proposal, we were focused on the funders and met with some folks in the bibliometrics community who recommended that we really focus on funders. So we went through these publications to try to identify who funded that. Specifically, we targeted the top 10 funders of biomedical research in the world today. I neglected to include that figure, but that's NIH, it's Medical Research Council, Wellcome Trust, Chinese National Science Foundation, Canada CIHR is in there. And so this is a graph of that. And again, they're all pretty comparable. One of the things that we noticed is that Howard Hughes seems to get much better data sharing than the other funders. So that's something interesting that we'll follow up on. We don't know if that's because of the requirements they put on it, or, Howard Hughes is famous in that they have a different funding model, that they fund the investigator and not the project. So maybe that says something. You choose people that are dedicated to science, then maybe you don't have to force them to be transparent about their data. We'll be chasing that down. So that's just something that we've been up to, or I've been up to for the past few months. I didn't have time to make a slide about what the rest of the team has been up to. Arsh and Eric have recently released the 2.0 release of the Healthy Volunteers data set. Mia and Dustin have been busy with a pre-registered project for the ABCD data that is it was an HBM abstract and will be soon be another paper. So those are the kinds of things going on. And oh, the last piece of this, sorry. Apologies. The absolute last deliverable is improving the metrics using large language models. Again, my fonts are off. So again, the stuff that's out there right now is using a regular expression approach. So we worked with John Lee to use LLMs to do this, to sort of ask the LLM to look for these things. We're using a structured JSON mode, which creates a Mongo compatible object mapper. Sorry, this is just gibberish. This isn't relevant from the previous time. So this is what we get. So when you look at the LLM metrics, which is in the red, versus the OddPub metrics, which is in the blue, you can see that we get more true positives, but we also get more false positives. So we are not yet in the space that we can get the LLM to do better than simpler approaches, but we're relatively early on. So we think this is still coming. All right, now it is 10:10, and I've told you everything I already tell you. Any other questions?